Yeah, okay. I will proceed. Good afternoon, students. Good afternoon. Good afternoon, everyone. Yes, good afternoon. In morning session, we have completed part 1. Okay, 60 question we have discussed. Now we are going to discuss the remaining questions of previous year 2022. Okay, be attentive in class. If you have any doubt, kindly let me know. Okay. First question, what they are asking? What type of DNA molecule? What type of DNA molecules are copied using rolling circle replication? Okay. They are asking which type of method, in which type of DNA molecules are copied using rolling circle replication process. Okay. If you see normal DNA, for example, eukaryotic DNA is there. In replication, what it will do? It will a bidirectional replication will be there. Okay. Bidirectional replication will be there. Okay, correct. This is correct. If the prokaryotic genome like circular genome also, that will be replicated by bidirectional method. But I already told in our previous class itself, bacteriophage lambda genome will have, specific lambda genome will have rolling circle method of DNA replication. So the right option will be option B. Okay, remaining what? Yeast chromosome, it is a eukaryotic. Mitochondrial genome, there also bidirectional method of replication will happen. Then bacterial chromosome, there also the same thing will happen. So rolling circle method will be happen in bacteriophage lambda. Okay. Second question, plasmid vector will essentially are. They are asking particular about plasmid vectors. Okay, we know plasmid vector means what? It's a double stranded DNA molecule that will have its own origin of replication. Okay, own origin of replication will be there. This is the first point. It will have its own origin of replication. Second thing, there will be a selection markers will be there. Okay, selectable markers. Particularly if you see antibiotic resistance genes will be there, or laxate genes will be there. Blue white screening we can do. Okay. Then third most important thing is what? Multiple cloning site will be there, because this a particular fragment will be removed, there our gene of interest will be cloned. So what will be the right answer? Origin of replication will be there for multiplication, selectable marker for selecting a recombinant, and multiple cloning site will be there for cloning our gene of interest into our vector. Okay. Then another option you see promoter, terminator sequence. These are specifically present in where? Expression vector. If a vector is expression vector, it will have a it should have the promoter sequence, terminator sequence, everything for production of that particular proteins. Okay, whatever gene we are cloning, that will be transcribed into messenger RNA, that will be synthesized into proteins. Okay, that is expression vector. Okay, that should have promoter and terminator regions. Okay, another question from genetic engineering. Genetic engineering involves the following steps. Okay, different steps are there. They are asking to arrange the steps in sequence. We know in genetic engineering, first we will what we will do. For example, the genomic DNA or whatever it there, this we will isolate the DNA. Then we will cut the DNA with restriction enzyme. So our target fragment will be there. This we will clone. This we will clone in our vector. Okay, this we will call as what? Construct. This construct will be what? Transformed in the E. coli. This also we know this will be transformed in the E. coli. Then after that what we will do? We will select the colonies, or we will select the bacterium that will having our gene of interest. So transform and selection will be there. So this is the basic step. So these are the basic steps. Based on that we have to arrange the things. First thing was gene isolation. Yeah, first C, wherever C is coming, that will be the right answer. First gene has to be isolated. Then manipulation and vector construction will be done. After that, what will be there? Transformation we will do. Then we will select and confirm the transform cells. Okay. So option D will be the right answer. Okay. C, A, D, B will be the right option. Okay. Okay. Yes. Sorry, yes. Next, they have given about the restriction enzyme, okay. Alkaline phosphatase, they are talking about enzymes, okay. Enzymes that are used in genetic manipulation. Particularly here, they have asked the function of alkaline phosphatase. Alkaline phosphatase is particularly used for what purpose? Alkaline phosphorus is particularly used for which purpose? For removing the phosphate group from 5' end of the DNA molecule. Okay. So, it will add phosphate group? No, it will not add. Add or more deoxyribonucleic. This also is not the function. Removing the phosphate group at 5' terminal of the DNA molecule. This will be the right answer. Okay. Now, which enzyme will add phosphate group to the DNA molecule? Yes, kinases. Kinases is the enzyme that will add phosphate group to the DNA molecules. Okay, clear? Which of the following restriction enzyme does produce fragments with blunt end? Okay, blunt end upon restriction digestion. They have given different restriction enzymes. We have to find what are the enzymes after recognizing that is cleaving the double stranded DNA into blunt ends. So, blunt ends will be produced. What are the restriction enzyme given here? Okay. We know BAM H1, it is a sticky end producing enzyme. This is one of the most commonly used enzyme for gene cloning. Okay. HIND3, this is also a restriction enzyme that will produce sticky end. Not one also produce the sticky end, but SMAM, SMA1, if you see, the recognition sequence will be CCG, GG. GGG will be there. It will cut like this. So, it will produce an blunt end. Clear? So, the right option will be SMAM1. Okay. Which of the following is not a millet? This is general agri question. This is one of the simplest question also. We know millets. Millets we can classify majorly into major millets and minor millets. Okay. This we know. They are asking which is not a millet. So, what among this is not a millet? Jowar, yes, it is a millet. Wheat, wheat is what? It's a cereal crop. Okay, ragi is millet, okay, bajra. So, the right option is wheat. Okay. Next, a transgenic event is differentiated from other events. Okay. How we will differentiate the transgenic event from other events? Okay. Copy number means what? For example, this is our transgene. How this transgene is integrated into the gene of interest. Okay, gene of interest. Copy number means how much multiple copies it has integrated. That is copy number. Site of integration, whether where in at which position it is integrated. These are not. These are called as what? Transgenic events. Okay. So type of protein produced. This will not come under a transgenic event. It's not a transgenic event. So both copy number and site of integration will comes under the transgenic agent. Okay. So this can be easily differentiated from the other events. Okay, clear? This is simple question only. Okay. Then another important question. Okay. See here about markers they are asking. Maximum number of type of alleles. Maximum number of type of alleles. A recombinant inbred line population. They have given an inbred line population. That means it will have homozygous. That will derive from an two contrasting rice genotype. Okay, two contrasting rice genotypes will be there. Okay, can generally show a SSR locus. They are asking how much SSR locus will be there. Okay, how much SSR allele will be either capital A or small a will be there. Only two alleles will be there. Okay, so what would be there? SSR here it will come biallelic. Okay, so the right option is 2. This is clear. This is simplest calculation only. You will do this one. Okay. Here so much of repeated questions are there. Okay, we will see that also. Okay. Plasmid DNA isolation method are primarily based on, okay, if, for example, this is our bacterium, it will have the genomic DNA also, then plasmid DNA also will be there. How to isolate the plasmid DNA? Okay, how to isolate the plasmid DNA from this genomic DNA? It will be primarily based on what? This will be not based on location and sequence. Restriction site also will not come. Yes, this size and conformation only. Okay. Whether it is covalently circular molecule. Okay. or supercoiled molecule. Based on these things, we will separate the plasmid DNA from our genomic DNA. Okay, clear? Tissue culture for plant transformation should be free from. They are saying tissue culture for plant transformation. Tissue culture means what? We are taking our explant, we are generating the plants from there. Okay, that should be free from what? First thing, high rate of shoot regeneration should be there. Yes, there should be a high rate of shoot regeneration. Root also should be regenerated more. Okay, this two are correct. Agrobacterium co-cultivation. Where comes this agrobacterium co-cultivation? If you see in transformation, we will treat our explant. For example, this is a explant. Okay, this explant will be co-cultivated with the agrobacterium strains. Okay, having our plasmid DNA. Okay, so co-cultivation we will do. This we will keep. These plants we will keep on media for generating of the plants. Okay, clear? So agrobacterium co-cultivation also will be there in plant transformation method. Okay. Somoclonal variation, that means what? Somoclonal variation means while developing these plants, whatever the variations happening due to chimeric genes or whatever these things, that is known as somoclonal variation. So which should be free from this? Somoclonal variation should be free from that one. Okay, that will cause us an undesirable phenotypes will be produced. For albinism phenotype like that will be produced. So somoclonal variation should be free from that tissue culture. Okay. Function of ligase, another simple question only. What is the function of ligase? It will ligate the double stranded DNA. TK2 double stranded DNAs can be ligated, or what it can do? If a double stranded DNA is there, if there is a missing of phosphodiester bond in one of the strand, this can also ligated by DNA ligase. Okay, you see now option to repair single stranded breaks that arise in double stranded DNA. Yes, it will make to join together two individual fragments of double stranded DNA. So both are correct. Okay, so both A and R are correct. Yes, this is simple question only. Okay, then you have to remember about the cofactor of this DNA ligases also. This is another repeated question. DNA ligase cofactor. Okay, if it is E. coli, the cofactor will be NAD plus. This we have already seen. If it is T4 bacteriophage, the cofactor will be ATP. This we already discussed in our previous lecture. This question they can ask. Okay, this is also another important thing. Okay, you have to remember. Agar, the primarily that will be used for what? Gel preparation, everything we are using. It's a gelling agent used for tissue culture media. So tissue culture, this is different game. Is obtained from which? Agarose will be used in agarose. This will be used in as a gelling agent for preparing our gel in gel electrophoresis. Okay, gel electrophoresis. Okay, for media preparation, tissue culture media preparation, we will use agar. Okay, that will be gelling agent for tissue culture media. That is obtained from where? From where? They are asking from where. This will be obtained from red algae, Gelidium amansi. This is 2019 JRF question also. Okay, 2019 JRF question also. Okay, this agar was isolated from red algae, Gelidium amansi. Okay, the name is Gelidium amansi. Okay. Suspension culture, this we have seen in morning session only. Suspension culture is generally used for what purpose? Generally, it will be used for production of metabolites, secondary metabolites. Yes, secondary metabolite production, we will use suspension culture. Haploid production, anther culture we will use. Okay, micro propagation, different explants we will take. We will cultivate in tissue culture. Okay, in vitro pollination, this will not come. Okay, in tissue culture. Okay, so the right option for suspension culture is what? Production of secondary metabloids. Yes. Which of the following is an example of protein domain? First, you have to know the difference between domain. What is meant by domain? What is meant by motif? Okay, this is another important question. Okay, they can ask in question exam. Okay, first you have to know, we know proteins means what is a sequence, like amino acids will be there, like this different amino acids are there. Okay, this will be what? The sequence of amino acid is known as primary sequence. Okay, if this sequence are arranged to form secondary sequence, okay, secondary structure will be formed like alpha helix, beta sheet, antiparallel, parallel beta sheets. This will form. Okay, this will be further folded to form tertiary structure. Okay, this we know this is tertiary structure or 3D structure. Biologically active structures will be formed. If one tertiary structure bind with another thing, this will be known as quaternary structure. This is simple thing. Okay, quaternary structure will be formed. Now we are going to see what is meant by domain and motif. Okay. If you remember fatty acid synthase enzyme complex, fatty acid synthase enzyme complex is a single polypeptide enzyme. It will have 7 different catalytic activity. Okay, 7 different catalytic activity will be there, but it is a single polypeptide enzyme. Okay, so domain means what? It's a individual or independent functionally domain, independent functionally region of an polypeptide. Okay, so this is an one domain. This is first domain. This is second domain. This is third domain. It will have, even though this segment will be removed, this first segment have the capability to do the catalysis. Okay, so that's why we are saying it's an independent functionally unit of an protein, that is known as domain. Okay, motif means what? For example, secondary structure are there, alpha and beta, like that. If this secondary structure are bind together, for example, alpha beta alpha, okay, this is one of the motif. Okay, this will bind, but this will not have an catalytic activity, or this will not have an independent functional unit will not be there. Okay, clear? So domain will function independently. Okay, motif is not an independent functional unit. Okay, this is clear. Now we are going to see the actual question. Which of the following is an example of protein domain? Okay, we know beta sheet, it's a secondary structure. Exon will never come here. Globin protein, here they have given about the entire protein. If you see zinc finger is a type of transcription factor. It will go and bind with the target DNA. So that will have an individual and independent activity. So zinc finger domain is the example of domain. Okay, clear? This is important point. Okay. What is an open reading frame? What is an open reading frame? If a double stranded DNA is there, if a double stranded DNA is there, okay, this will be transcribed to mRNA. This mRNA will be translated into proteins. Okay. So here codons will be there. Okay. Codons means what? It's a triplet of nucleotides. Okay. Triplets of nucleotides that will code for amino acid. Okay. There will be different open reading frames will be there. Okay. Either, for example, ACC is there. If this started with first one, here one open reading frame will form. If the C will be considered as a first nucleotide, second open reading frame will form. If we consider this third C as a first nucleotide, here different open reading frame will form. So open reading frame means what ultimately? It's a nucleotide of a gene that make up the codon specifying the amino acid. So how it is specifying the amino acid, that is known as what? Open reading frame. I hope this is clear for everyone. Okay. Isoelectric point, another important and simple question they are asking. The isoelectric point of a protein is defined as what? What is meant by isoelectric point? We have already discussed, it is the pH at which protein will have net charge of 0, or the mobility of the protein will be 0. Okay. So the pH at which the protein has no net charge is known as isoelectric point. Yes, very good. Option A. Option A will be the right answer. Hot star PCR. While our PCR variant class itself, I have told hot star PCR involves the following step. Okay, I told the enzymes, okay, they will use here the enzymes that will be activated only at particular temperature, and there will be a changing of temperature particularly for specific amplification. Okay, this I told during our class itself. Now you have to match. What will be the answer? First, adding PCR primer. Yes, first we have to add the PCR primers, buffers, DNTPs, template, everything. Okay. Next, what we will add? So here we can easily, by after seeing the first option itself, we can easily eliminate the other things. So first A only should come. Okay, first A only should come. Second, what? Attaining reaction temperature. Then we will add the polymerase. Then continuing PCR to amplify the specific amplicons. Okay, we can easily eliminate other things. Okay, so option B will be the right. Yes, very good, very good. A, C, B, D. Yes. Next question. They have given here the different cloning vectors. Different cloning vectors they have given. Okay, we have to match with the following. Okay, match and let me know the answer. PBR 322, Plasmid University of California, PGEM 3Z and M13 MP1. What will be the answer? Yes. PBR 322 is a specific example and it is the first vector ticket designed. Okay. It is a E. coli plasmid based vector to clone DNA. Yes. First will come option 3. Easily we can eliminate remaining these 3 options. So first will come only one option have. First as option 3. Plasmid University of California is a what? Is a lag selection strategy will be there. This is also clear. Lag selection will be there. Recombinant will produce white colony. Non-recombinant will produce blue colony. Okay, this is JRF 2019 question also. Okay. Okay. PGM3Z, what it will do? It's a cloning vector based on, it's a vector facilitating in vitro transcription. Okay. M13 MP1 cloning vector that will be based on bacteriophage. Okay, clear? I hope this is clear for everyone. This is simple question. You will answer. Okay. Then this year they have asked 4 questions from DNA modification enzymes. Okay, DNA modifying enzymes. Okay, another question from exonuclease. Okay, ball 31 exonuclease and lambda 3 exonuclease. About they have asked this. We have covered in our class itself. Okay, BAL 31 exonuclease removes the nucleotide from both the strand of the double stranded DNA. This while taking lecture itself, I told this question they will ask. Okay, BAL 31 is a specific exonuclease. It will remove the nucleotide from both the end of the double stranded DNA molecule. Okay, from both the end, it will remove the nucleotides. E. coli exonuclease 3, I told 3 means it will remove the nucleotide from 3' end of the DNA. Okay. So it will remove nucleotides from 3' end. So from both the strand of double stranded DNA. Yes, this is also correct. So both statement 1 and statement 2 are correct. Yes, very good. Both statement 1 and statement 2 are correct. Okay, clear? Both statement 1 and statement 2 are correct. Any doubt in this? Yes. Codon anticodon interaction. We know codon anticodon interaction. We know mRNA is there. Take care. This will have an nucleotides will be there. This we will say as codon. Take care. This will be binded with a tRNA molecule by codon anticodon. How this interaction will happen? Whether it will happen with electrostatic interaction, covalent bonding, hydrogen bonding, or hydrophobic interaction. So it will happen by hydrogen bonding, base pairing between the purine and pyrimidines. Okay, so hydrogen bonding will be happen between codon and anticodon interaction. The region of the soil around the root surface. Simple question. Rhizosphere is the region. This we know. This is very simple question. Everyone will write very simple question. The region of soil around the root surface is rhizosphere. Okay. The energy required to convert one molecule of liquid water to one molecule of water vapor. One molecule of liquid water is converted into one molecule of what vapor. Okay. What? Yes, very good. Heat of vaporization. We are converting the liquid to gaseous form. Okay. Heat of vaporization. This is another important question they have asked in 2024 also. Repeated question. Exudation of sap from the cut surface of the stem of Wells watered herbaceous plant is due to positive pressure in the xylem, which is termed as what will be the right answer? Exudation of sap from the cut surface of the stem, well watered herbaceous plant is due to, yes, very good. It will be due to root pressure. This we have already discussed. Because of root pressure only, this exudation will come. Okay, very good. Which one of the following is not involved in photosynthetic electron transport process? We know electron transport will be happen PS2 to PS1. From here NADPH2 will be synthesized. Okay. Here photons will be there. Okay. Photolysis of water will happen here. Now they are asking which is not involved in photosynthetic electron transport. Excellent. ATP synthase will not be involved. Okay. Photosystem 1, photosystem 2 will be involved. Phytin will be there and plastoquinin will be there. Apart from that, cytochrome B6F also will be there. Very good. In metabolically active cells, this also they have asked in 2024. In metabolically active cell, the osmotic pressure is equal to osmotic pressure is equal to which component? Osmotic pressure. Yes, osmotic pressure will be equivalent to which component? Pressure potential, solid potential, matrix or water potential? Excellent. It will be equivalent to solute potential. Very good answer. Okay. It will be equivalent to solid potential. Another thing you have to remember. Okay. This osmotic pressure and solid potential will have same only. That means that will be equal only. But this will have opposite sign. Okay. Osmotic pressure will be in positive. Osmotic potential will be there in negative. Okay. Clear. This is also another important point you have to be remember. Okay. Keep remember. Okay. The membrane protein channel or pores controlling the selective movement of water. Water channel. They are asking about water channel. There is a membrane protein. Okay, or channel or pores that is controlling the selective movement of water is what? Whether it is a aquaporine or channel protein, carrier protein or transport protein. What will be the right answer? Students, what will be the right answer? What will be the right answer? Yes, I am waiting for your answer. What will be the right answer? Yes, it's an aquaporine. TK. Aquaporine is also known as water channel. TK. TK. That's why I'm writing here. Aquaporine is also known as water channel. TK. Peter Ager got Nobel Prize in 2003 for this discovery. TK. Aquaporine. Water channel is known as aquaporine. That's why I have written here. TK. Peter Ager got Nobel Prize in 2003 for aquaporine. This is 2025 ARS question. TK. They have asked this time in prelims. TK. Clear? This you will answer. I know this is very simple question. Kindly match and let me know. Different scientist name they have given and order of discovery. Who has discovered which hormone? This is 2023 question also. Okay, they have asked 2023 same question. So much of questions are repeated in this paper. Okay, what will be the answer? Choose the correct answer. FW went. Yes, oxine. Auxin he has discovered. Kuraswo, he has discovered gibberellin. Yes, 2 will come option B. Okay. Skoog, cytokinin, Waring and Adicot, abscisic acid. Okay. So what will be the answer? 4, 3, 2 and 1. Okay. Option A is the right answer. Yes, 4, 3 and 2, 1. The regulation of plant development by light is what? Okay, you have to know what the regulation of plant development by light is termed as. Another question I am writing, the regulation of flowering by light is known as what? Flowering is what? The regulation of plant development by light is termed as what? Photosynthesis, differentiation, photomorphogenesis and photorespiration. Yes, very good. Photomorphogenesis. Okay. If the light duration and light is affecting the flowering, then how it is regulating the flowering? That is known as photoperiodism. Okay. You have, then again I am saying, you have to keep remember who has given the term photoperiodism, vernalization, okay, Garner and Allard, okay, photoperiodism, vernalization by T.D. Lysenko, okay. These are important scientist name you have to keep remember, okay. This is very important point, okay. Natural rubber is a what? It from secondary metabolites. From last 4 years, only one secondary metabolite question they are asking. Okay. But secondary metabolites is one of the important topic. Here they are asking natural rubber is an polymer of what? It's a polymer of isopentenyl pyrophosphate. Okay. It's a polymer of isopentenyl. Yes, very good. It's an isopentenyl unit. Polymer of isopentenyl unit. Okay. Very good. The seeds requiring light for induction of germination process is called as, whether it's an positively photoblastic seed or negatively photoblastic or long day or short day. What will be the right answer? Yes, example we can say lettuce. Yes, it's a positively photoblastic seed. Okay. Example is lettuce that will requires light for induction of germination process. Okay. Okay, clear. The precursor of ethylene synthesis in plant. What will be the precursor for ethylene biosynthesis in plant? Precursor for ethylene biosynthesis in plant. Tryptophan, methionine, indole, and kaurin. Yes, very good. Methionine, yes. Methionine for ethylene. Tryptophan for auxin. Indole also will come for auxin. Tryptophan independent pathway. This is tryptophan independent pathway. Okay. Then another thing is what? Ent kaurin. This will come in gibberellic acid synthesis. Clear? So the right option is what? Methionine. Okay. Yes, very good. The principal function of light dependent reaction of photosynthesis is to generate. Light reaction will always generate what? ATP and NADPH2. This is the clear. This is simple question only. ATP and NADPH. Okay. Yes, this will be the right option. In plants, the tubular extension of plasma membrane that connect the cytoplasm of adjacent cell. For example, two cell is there. The tubular extension of plasma membrane that is connecting the cytoplasm of the other cell is known as what? It is what? It's a plasmodesmata. It's an endomembrane system, plasmodesmata. Yes, very good. This we have already seen. The breakdown product of DNA, this you will answer. This they have asked 2025, 2024 and 2022. 3 times repeated question. Breakdown product of DNA capable of inducing cell division, which was later identified as cytokinin. Yes, excellent. Kinetin. Okay, that is the breakdown product of DNA. Trans-cytidine benzylarenine, these are what? These are synthetic one. This thiogyron is also synthetic cytokinin. This will be used as a defoliant. At high concentration, we can use this as defoliant. The amount of carbon dioxide assimilated, amount of carbon dioxide assimilated, section 1, shift 1, shift 2, 2022. The amount of carbon dioxide assimilated by photosynthesis divided by amount of water transpired is what? Water use efficiency. You see, every year they are asking the same question. 2025, 24, 23 shift 1, 23 shift 2 and 2022. Transpiration efficiency and water use efficiency. From this, sure they are asking one question. Okay. Growth parameter analysis, from there, sure they are asking one question. You have to be very strong in that. Okay. If you want to prepare, if you want to work like topic wise objectives, you can kindly enroll in this mock test series. Okay. Then those who are preparing for 2027 ICAR examination for plant biotechnology, they can enroll in our master course. Okay. This will be very useful for your preparation. First 100 students will get offers also. Okay. Kindly enroll and prepare well. Okay. Next question. Which of the following is correctly matched? 2024, the same question they are asked. This we can easily skip also. Sugar cane, yes, Cran's anatomy will be there. Pineapple is not a C4 plant, it's a camp plant. So wherever B is coming, that will not be answer. Yes. Rice is a C3 plant, yes. Groundnut, yes. So A, C and D will be the right answer. Very good. The organelles involved in photorespiration. Very simple question. CPM, chloroplast, peroxisome and mitochondria. Yes, CPM. So option B, chloroplast, mitochondria and peroxisome. Very, very simple question. Yes. Yes, very good, very good. Option B. In mitochondria, the enzymes of Krebs cycle will be present where? It will be present in matrix. Yes, in the matrix. Okay. It will present in mitochondrial matrix. Okay. Students, you have to remember here properly. The organelles involved in process of photorespiration is chloroplast, peroxisome and mitochondria. Okay. You have to remember the sequence also. First chloroplast, next peroxisome, next only mitochondria will come. Okay. This is very important. They can ask this sequence also. Clear, students? Pressure potential in the xylem of actively transpiring cell. This you have to answer. This we have already discussed in 2023 question paper. Pressure potential in the xylem of an actively transpiring plant is whether it will be 0, negative, positive or minus 0.1 megapascal. What will be the answer? Whether it will be 0, negative, positive or 0.1 megapascal. 0. No, it will not be 0. 0 potential will be there for only for pure water. Okay, that is the maximum water potential. This we have already discussed. They are asking about pressure potential. Pressure potential, for example, if a cell is there, the water has to come inside the cell. It should have a positive potential to inside it, positive potential for entering inside the cell. If it is negative potential, means it has to be involved for transpiration. Okay, for transpiration. Okay, for transpiration, this should have negative potential. Clear, students? So yes, very good. Negative potential should be there. If the water has to come inside the cell, positive potential should be there. Yes. The world leading exporter of sunflower seed. General agri question. The world leading exporter of sunflower seed. Okay. Sunflower seed oil. Okay. Ukraine. Russia. Now Russia is the leading producer. Okay. Now Russia. Okay. 2022 Ukraine. Okay. Now Russia is the leading producer. Okay. leading producer come exporter. Okay. Another important thing, India is the largest importer. Okay. Importer of soybean oil. Okay. Sorry, sunflower seed oil. Based on the water use efficiency, identify the sequence of crop in ascending order. They are saying, question itself, ascending order, okay, from smaller to higher. Which will have the higher water use efficiency, C3, CAM or C4? We know CAM will have higher water use efficiency. So CAM should come at last. Only in these two options, CAM is coming at last. Okay. C3 plant will have less water use efficiency comparing to C4. So C3 plant should come first. Green gram, finger millet. Yes, this will be the right option because wheat is a C3 crop. This will also not come. Clear. Just we are selecting the option by elimination method. Okay. So green gram, finger millet and pineapple will be the right answer. Another important question. Every year they are asking the flow of electron. Flow of electron in electron transport chain of photosynthesis. Okay. From where to where the electron will be transferred. This you guys have to answer. Identify the current sequence of electron flow leading to ATP synthesis in cyclic photophosphorylation. In cyclic photophosphorylation, what will be the sequence of electron? You have to answer now. Cyclic means it will start, it will, wherever it is starting, that place only it will end. Okay, first thing, that one only. Okay. In two option, nothing is there. So this we can easily eliminate. Yes. Second thing, you see, plastocyanin. It's not a primary electron acceptor from, it's not a primary electron acceptor from P700 because from P700, I told in the yesterday class itself, phyllocyanone or A0 or A1 is the primary acceptor. If PS2, that means what? P680 means primary electron is accepted by pheophytene. This also I have told pheophytene will be there. So what will be the right answer? From P700, electron will be accepted by A0, A1. That will be transferred to iron sulfur clusters, then to ferredoxine. But it will be cycled back by cytochrome B6F, then cycled back to P700. Clear? So option D will be the right answer. Clear? Uday Lakshmi, clear? Yes, I hope this is clear for everyone. TK. TK. This is very simple question. Don't do mistake. This is very simple question. Read the question properly and pick the answer. Okay. Identify the sequence of nutrient element. Sequence of nutrient element in crop capable of getting redistributed. If an element is redistributed under deficiency condition, it should be a what? It should be a mobile element. That then only it can be redistributed. They are asking in ascending order. This is 2024 question. We have already discussed. What will be the answer? I am checking how you are listening the class. What will be the answer? Yes, very good. Okay, nitrogen is more mobile. Okay, boron is immobile. So the right option will be B, and right option will be D. Yes. An example of short day plant. What will be the example for short day plant? Boron, sorry, what will be the example for short day plant? Spinach, barley, henbane, cucumber. This is a day neutral plant. Typical example of day neutral plant. What will be the example for short day plant? Any guesses? Spinach, barley, henbane, cucumber. Cucumber will not come because that is a day neutral plant. Yes, barley, henbane, spinach. What will be the answer? Yes, spinach. Okay, that is long day plant. Henbane, long day. So barley, TK, wheat, barley, TK. Abscisic acid synthesis in plant involves what? Abscisic acid synthesis in plant involves isoprenoid pathway, SAM pathway, carotenoid pathway, IAA pathway. This is another simple question. Abscisic acid pathway in plant involves. We know Indole acetaldehyde pathway will be involved in IAA synthesis. So wherever D is coming, that will not be answer. Easily we can eliminate the options. Okay. Isoprene pathway, yes, it will come. Carotenoid pathway, yes, it will come. SAM pathway for typically that will involve for ethylene biosynthesis. Correct, ethylene biosynthesis. So A and C will be the right option. Clear? So A and C will be the right option. Methyl vialogen, this we have already discussed. Methyl vialogen is also known as paraquat. Okay, used as a herbicide that will inhibit the light reaction of photosynthesis. In our class itself, we have discussed. Yes, this is true only. Statement 2. Paraquat blocks the electron transport chain at plastoquinone of PS2. Whether it will inhibit the plastoquinone of PS2? Any guesses whether it will inhibit the plastoquinone of PS2? It will inhibit PS1. I am right. It will inhibit the PS1. Okay, so what will be the right answer? So statement 1 is what? Right. Statement 2 is wrong. Statement 1 is correct and statement 2 is wrong. What will be the right answer for this question? Uday Lakshmi, just solve and let me know. Uday Lakshmi is asking some doubts. Cucumber is day neutral plant. Okay. What will be the spinach is short day or long day? Yes, let me know. Spinach is short day or long day? Spinach is long day plant. Yes, very good. Then henbane and barley. Okay, we have told wrong. Okay, henbane will be the right answer. Barley was long day plant. Okay. Okay. I hope this is clear now. Okay. Now next question. The essential cofactor involved in water oxidation. The essential cofactor. The essential cofactor involved in water oxidation. Essential cofactor involved in water oxidation is what? Manganese, chlorine. Okay, manganese and chlorine will be there involved in water oxidation cycle. This we have already seen. Okay, magnesium will be involved in chlorophyll. Okay, this is clear. Copper will be there in plastocyanin. Clear. So read the question properly. The essential cofactor in water oxidation process. Water oxidation process. What is the essential cofactor like that? They are asking. It is manganese. Okay, manganese. Okay, clear. In acid soils at pH 5, the element that become available in toxic amount. Okay, there we know that optimum pH will be if 6 to 6.5 is there, optimum pH for maximum absorption of mineral elements. Yes, this is clear. They are saying if the pH is less than 5, if the pH is become less than 5, what will happen? Which element will be available as a toxic form? We know aluminium will become available at more acidic pH. So if pH is less than 5, aluminum availability will be more, that will be uptaken by plant, osmotic stress will happen in the plant. Okay, then high metal toxicity also will happen. Phytochrome is localized within. This is already discussed question only. Okay, cytosol, this is already discussed one. Bacane disease in rice is associated with discovery of which plant hormone? This you guys have to answer. This will be associated with which type of plant hormone? Gibberellic acid, ethylene, cytokinin. This we can also say as foolish seedling. Yes, very good. Gibberellic acid. When tissue culture medium contains both auxin and cytokinin in the ratio of 10:1, the pith cell of tobacco grow and form callus. Further, They are saying the ratio is 10:1. If the ratio of auxin to cytokinin is less in the medium, a number of roots are initiated from the callus. They are saying if the ratio of auxin to cytokinin is less, auxin to cytokinin is less means root is forming, like that they are saying. In statement 2, what they are saying, if the ratio is more, for example, cytokinin is less than auxin, a number of shoot buds are initiated, like that they are saying. What will be the right answer? This is another simple question. What will be the right answer? Yes, you have to answer. Ratio of cytokinin to auxin. This we have already discussed. What will be the right answer? Both are wrong. Very good. Okay, both statement 1 and 2 are wrong because high auxin to cytokinin ratio only will promote root. High cytokinin to auxin ratio will promote shoot formation from callus. This we have already discussed. Yes, so both are wrong statement. Another important question and very simple also. Short-term treatment for about 16 to 20 hours given to cut flowers with high concentration of sucrose. Whether I am audible now? Yes. Kindly reply whether I am audible. Kindly reply yes or no whether I am audible now. Yes, yes. Thank you, thank you. See, this we have seen. Okay, a short-term treatment of 16 to 20 hours. Okay, of this chemical substitute if you give means what is that process? That process is known as pulsing. Why we are using only this component? I just want to give the explanation. Sucrose will act as a carbohydrate source for the respiratory substrate. Okay, silver nitrate will inhibit the action of this ethylene. Okay, benzyl adenine. Okay, this will it will decrease the activity of ethylene. Okay, so for this purpose we are including these components. Okay, another important question from mineral nutrient. They have given the different mineral elements. We have to match with the physiological functions. Okay, physiological functions they are asking. Calcium, yes, we know this is particularly involved in what? Chromosomal components will have this calcium because negative charge will be there in the phosphate group of DNA. This can chelate, this will be bind with the calcium. So, first we'll have what? First we'll have option 5. Okay. So, A, only one option is coming 5. I am right. So, only one thing is coming 5. So, B, zinc. It's a constitute of superoxide dismutase. Yes. 5142, very good. 5142, yes, this we have discussed. Sulfur, it's a constitute of glutathione. Yes, this is also we know. Magnesium is involved in activation of rubisco carboxylase. Yes, 5412 will be the right option. Very good. Another important question from senescence. Senescence, they have given the different types of senescence and they have given the example of the crops. Okay, just we have to match. They have given the different types of senescence. Overall senescence, progressive, top senescence and deciduous senescence. We have to match with the particular example of the crop. Anyone can match. Overall senescence, that means the entire plant will be died there, correct? Entire plant, root, shoot, everything will be completely senescence. Okay. Top senescence means what? Only above ground parts will be senescence, but below ground part will remain okay active only. So, deciduous senescence and progressive senescence. You have to answer now. Okay, this option is wrong. Okay. Okay. So, overall senescence will be there in wheat. This is very simple question. Yes. Both the root as well as in shoot, everything will be died after the completion of the life cycle. So, overall senescence will be there in wheat. Progressive senescence, particular example is lemna. Okay, clear? Lemna. Yes, B is the right answer. Yes, B is the right answer. Okay. Top senescence in sugar beet, the root, that means the tubers will be remains active only. The shoot will be senescent. So, C will come option 1. Deciduous senescent example is gulmora. Okay, gulmor. Clear, students? Most widely used chemical for sugarcane ripening. You have now itself I am saying read the question properly. They are asking the function of sugarcane ripening. Okay, they are not asking about internode elongation like that. They are asking about sugarcane ripening process. What are the hormones will be involved? Yes. Glyphosate is what? It's an herbicide. How we will use the herbicide for ripening? So wherever D is coming, this will not come. Yes, correct. Ethyl is one of the ripening hormone. Ethylene is ripening hormone. Ethyl is what? It's the trade name. Okay, commercially available trade name. Okay. So wherever A is coming, this will come. How salicylic acid will be involved in sugar, like sugar cane ripening? Simply it will inhibit the bacterial and whatever the abiotic, sorry, whatever the biotic organism infection is there now, that will be limited because of the ripening process can be happened soon. GA3, what it will do? Internode elongation can be done. Clear? So you have to read the question properly. Yes, very good. A and C will be the right answer. Very good, very good. Identify the recalcitrant seed. Every year they are asking one question. Example of recalcitrant seed. What will be the right answer? Recalcitrant seed. Mung bean, rubber, tea, wheat. This is simple question. We can easily eliminate the options and we can tell yes. Which is the example of recalcitrant seed? Okay, recalcitrant seed and orthodox seeds. Two types of seeds are there that we have already discussed. Okay, what will be the example of recalcitrant? Recalcitrant means desiccation, intolerant or tolerant. Kindly match and let me know the answer. Yes, I am waiting for your answer only. What will be the right answer for this? Recalcitrant seed, example. Orthodox seed, example. What will be the typical example for recalcitrant? Yes. Rubber and tea, recalcitrant. Mung bean and wheat, orthodox is. Okay, this is very simple question. Okay. Next, example of climacitric fruit. They are asking example for climacitric fruit. Okay. Which is not a climacitric fruit among this one. So, grape is not a climacitric fruit. Wherever option B is coming, that will not come as answer. Okay. Grape, citrus, these are what? These are non-climacitric fruit, correct? So, A, C and D will be the right option. Very good, very good, very good. The compound discovered as a natural trigger for metabolic explosion that raises the temperature of thermogenic inflorescence of Arium lily is what? This is another simple question only. TK, you will answer, I know. Yes. The compound discovered as a natural trigger for metabolic explosion that raises the temperature of thermogenic inflorescence of araminium. Ethylene doesn't have any role in thermogenesis. Abscisic acid also no. Brasnell steroid also will not have a direct role. Salicylic acid will be the right option. Yes, we can select these options by elimination method. Which of the following are found translocated abundantly in phloem? Yes, very good. Which of the following is transpirate abundantly in phloem? What will be the answer? Amino acids. Yes, amino acids will be transported. Yes, sucrose. Yes, these two are major thing. Apart from that, what it will come? If you see, potassium is another important element that will be accumulated more in phloem sap. Okay. So, A, B and D, where is coming? A, B and D. This will be the right answer. Yes. Okay. Identify the growth retardants. They are saying here growth retardants. Okay. Retardant means what? The growth will be inhibited. Okay. The growth is inhibited. Okay. They are asking which can be used as a growth retardant in crop plants. Now I want to give the explanation. See, malic hydroside, this will be primarily used to completely inhibit the sprouting. Completely inhibition of sprouting in potato tubers. Okay. Potato tubers. If you store the potato tubers for long times, that will be sprouted. Yes, that will be sprouted. So, this will be completely used for what purpose? To completely inhibit that tuber sprouting. Okay. Chloramate chloride, paclobutrazole, this both are what? Gibberellic acid inhibitor. I am right. These both are gibberellic acid biosynthesis inhibitor. Biosynthesis inhibitor. So, if gibberellic acid biosynthesis inhibitor means plant growth can be retarded. So, B and C ultimately it will come. Okay. So, wherever B and C is coming, that option will be right. Okay. Then second thing, we have to know whether D will come or not. D is what? Triiodobenzoic acid. TIBA is what? It's an polar oxine transport inhibitor. If oxine transport is inhibited, cell division will be stopped there. Cell elongation also will be affected. So, the plant will become stunted. So, this also we can use as growth retardant. So, B, C and D will be the right answer. Any doubt in this? B, C and D will be the right answer. Okay. You can practice Topic wise mock text also kindly enroll in our mock test series. Okay, those who are preparing for ICAR PG 2027 examination, kindly enroll in this plant biotechnology course. You will get 100% guidance from our academy. Okay, any doubt is there, students? Any doubt is there? If no other doubt means we can end the session now. Any doubt is there? Yes. No doubts. Yes. Thank you. Thank you, students. I will see in the next session. Thank you.